The 2024 Nobel Prize in Physiology or Medicine has been awarded to Victor Ambros and Gary Ruvkun for the discovery of microRNA and its role in post-transcriptional gene regulation. Scientific background for the discovery of microRNA and its role in post-transcriptional gene regulation. Evolution of multicellular organisms from unicellular ancestors, where each cell type acquired specialized functions, required increasingly sophisticated mechanisms of gene regulation. Besides transcriptional gene regulation, mediated by DNA binding, factors acting on regulatory sequences, other forms of control systems emerged as organisms with increasing complexity evolved over hundreds of millions of years. Genes encoding tiny noncoding RNA molecules, so-called microRNAs, expanded within genomes of multicellular organisms to exert post-transcriptional control over mRNA, stability, and protein translation. microRNAs and their mode of gene regulation remained completely unknown until the discovery by Victor Ambros and Gary Ruvkun in 1993. The two Nobel laureates investigated mutant, elegans nematodes with developmental defects caused by alterations at the Lin4 and Lin14 genetic loci. Ambros's laboratory cloned the Lin4 gene and made the surprising discovery that it did not code for a protein. Instead, it encoded a short 22 nucleotide noncoding RNA in parallel. Ruvkun's laboratory determined that Lin4 regulates LAN14 via multiple elements in its 3 and translated region 3TR. Upon comparing sequence information, they defined partial sequence complementarity between the short noncoding Lin4 RNA and the 3' UTR elements of Lin14. This provided a first glimpse into a conceptually novel type of regulatory RNAs, microRNAs. In 2000, Ruvkun's lab uncovered the highly conserved Let7 microRNA, leading to the subsequent identification of homologous microRNAs across diverse animal species, including humans. This sparked intense cloning and sequencing efforts to identify microRNAs across the animal kingdom, which led to the finding that microRNAs encompass a large group of regulators that control vast networks of protein coding genes. The discovery by Ambros and Ruvkun was completely unexpected and unveiled an evolutionarily conserved post-transcriptional regulatory mechanism mediated by microRNAs, with critical roles in animal development and for adult tissue function. Introduction. The control of when and where each gene should be transcribed into RNA and translated into protein is a fundamental aspect of life. For instance, insulin is produced in the beta cells of the pancreatic islets, whereas opsins are produced in the retina of the eye. Instructions for precise cell type specific gene regulation are encoded in the genetic material itself and acted upon by sequence-specific DNA binding proteins. Francois Jacob and Jacques Monod were awarded the Nobel Prize in Physiology or Medicine in 1965 for discovering how genes are regulated. The repertoire of DNA binding transcription factors is well conserved among unicellular and multicellular eukaryotes. King et al. 2008, whereas additional layers of gene regulation have emerged within multicellular organisms to ensure the correct production of RNA and proteins at any given time in each cell type. Regulation of cell type specific functions. Every cell contains an identical set of chromosomes and therefore the exact same set of genes. Cell type specific functions arise when only a select subset of these genes is activated within each cell type. Eukaryotic model organisms have been invaluable for genetic research, yielding numerous unexpected discoveries. Sidney Brenner introduced the nematode worm, Caenorhabditis elegans C. elegans, over 5 decades ago. This organism's short generation time, transparency, and ease of genetic manipulation have facilitated extensive study. Sidney Brenner, John Sulston, and Robert Horvitz used C. elegans to unravel how cell division, differentiation, and cell death are genetically controlled during organ development, discoveries for which they were awarded the 2002 Nobel Prize in Physiology or Medicine. In the 1970s, mutagenesis screens in C. elegans performed in the Brenner lab uncovered the Lin4 mutant E912. These worms displayed a striking phenotype. Many cell types and morphological structures were entirely absent, and eggs accumulated due to a failure in vulva. Development Figure 2. Horvitz and Sulston. 1980. Chelfie, Horvitz, and Sulston. 1981. Seemingly from the reiteration of developmental programs for specific cell lineages, the major disruption of worm development observed in the Lin4 mutant suggested that Lin4 encoded a master regulator of developmental timing. A large number of additional heterochronic mutants exhibiting various temporal developmental defects were characterized, including a second mutant, Lin14, discovered in the Horvitz lab. Ferguson, Sternberg and Horvitz, 1987. Heterochronic worm mutants with developmental defects. Nematode Lin4 and Lin14. Mutants with disrupted animal development. Mutant Lin for worms reiterate developmental programs for cell lineages to accumulate internal eggs without forming a vulva. While Lin14 mutants are small and lack larval development, worms adapted from Ambros. 2008. Meanwhile, Victor Ambros joined the Horvitz lab following his PhD with David Baltimore on poliovirus genome structure and replication. As a postdoctoral fellow, Ambros immediately embarked on genetic analyses of heterochronic mutants and identified Lin14 as having developmental timing defects that were opposite to those observed in the Lin4 mutant. Figure 2. In Lin14 mutants, larval programs were completely absent. Ambros and Horvitz. 1984. Notably, Ambros later discovered that Lin4 was a negative regulator of Lin14. Ambros. 1989. During this period, Gary Ruvkun had completed his PhD in bacterial genetics under Frederick Ausubel's supervision. While traveling through Europe, he became intrigued by worm genetics after learning about cell lineage analyses of heterochronic mutants. Chelfie, Horvitz, and Sulston. 1981. Ruvkun, Whiteman, and Ha. 2004. Subsequent discussions with Martin Chelfie and Robert Horvitz further fueled his interest in using C. elegans to investigate these questions. In 1982, Ruvkun started his postdoctoral research jointly between the labs of Walter Gilbert and Robert Horvitz, discovery of post-transcriptional gene regulation through microRNAs. In the Horvitz lab, Ambros and Ruvkun started their long pursuit to clone Lin14. At that time, identifying the DNA sequence for a locus defined by genetics was a challenging task. After years of persistent experimentation, they successfully identified the region using a classical restriction fragment length polymorphism approach. Ruvkun et al. 1989. During this period, both Ambros and Ruvkun obtained faculty positions. Ambros at Harvard University and Ruvkun at Massachusetts General Hospital and Harvard Medical School. Committed to their questions, they continued their molecular analyses. Ruvkun demonstrated that Lin14 is a nuclear protein with stage-specific expression in development, high in the L1 stage, and altered in Lin4 and Lin14 mutants. Ruvkun and Justo. 1989. Interestingly, Lin214 gain-of function mutants with deletions in the 3 UTR were discovered. Ruvkun and Justo. 1989. Whiteman et al. 1991. Leading to prolonged Lin14 protein detection beyond the L1 stage. Arasu, Whiteman and Ruvkun. 1991. Whiteman et al. 1991. The disruption of 3' UTR elements had no impact on the protein sequence, and Ruvkun, therefore, postulated that a post-transcriptional mechanism acting on mRNA stability, nuclear export, or translation likely mediated the temporal switch in Lin14 Whiteman et al. 1991. In contrast to the several Lin14 mutants identified, only one mutant in Lin4 had been found, E912. The Ambros lab set out to clone the Lin4 gene guided by restriction, fragment length polymorphism, and southern blot probing, walking along the chromosome, and iteratively testing smaller genomic fragments for their ability to rescue the mutant Lin4 phenotype. They pinpointed a 693 BP cell L restriction enzyme fragment. After rounds of open reading frame predictions and clone resequencing to rule out errors, they began suspecting that the Lin4 gene might be a non-coding RNA due to its short open reading frame off sequence. Frameshift mutations introduced into the C. elegans sequence did not affect Lin4 function, confirming this suspicion. In 1991, the lab began probing the Lin4 transcript by northern blot and RNase protection assay, revealing to short RNA transcripts of 61 and 22 nucleotides NT in length. Identification of two short Lin4 transcripts, northern blot of total RNA from wild type, LIN4E91 to mutant, and Lin4E91 to mutant rescued with cell I fragment, probed with radiolabeled Lin4RNA probe, compared to U6 loading control. Lee, Feinbaum, and Ambros, 1993. Having independently deduced the sequences of both Lin4, Ambros lab and LIN14 Ruvkun lab. On the evening of June 11, 1992, Ambros and Ruvkun exchanged sequence data for the Lin4 and Lin14 genes. Both noticed conspicuous partial complementarity between the Lin4 non-coding RNA and multiple elements in the Lin14 3TR. Figure 4. Recognizing the significance of their observation, the two labs performed an array of additional experiments, demonstrating that the Lin4 microRNA regulates the Lin14 mRNA through base pairing with elements located in the 3 UTR. Their seminal discovery was reported in two papers published back-to-back in Cell in 1993. Lee, Feinbaum, and Ambros, 1993. Whiteman, and Ruvkun, 1993. In the 1960s, it was shown that specialized proteins, known as transcription factors, can bind to specific regions in DNA and control the flow of genetic information by determining which mRNAs are produced. Since then, thousands of transcription factors have been identified, and for a long time it was believed that the main principles of gene regulation had been solved. However, in 1993, this year's Nobel laureates published unexpected findings describing a new level of gene regulation, which turned out to be highly significant and conserved. Throughout evolution.Ambros and Ruvkun were interested in genes that control the timing of activation of different genetic programs, ensuring that various cell types develop at the right time. They studied two mutant strains of worms, Lin24 and Lin14, that displayed defects in the timing of activation of genetic programs during development. The laureates wanted to identify the mutated genes and understand their function. Ambros had previously shown that the Lin4 gene appeared to be a negative regulator of the Lin14 gene. However, how the Lin14 activity was blocked was unknown. Ambros and Ruvkun were intrigued by these mutants and their potential relationship and set out to resolve these mysteries. Elegans is a useful model organism for understanding how different cell types develop. Ambros and Ruvkun studied the Lin4 and Lin14 mutants. Ambros had shown that Lin4 appeared to be a negative regulator of Lin14. Ambros discovered that the Lin4 gene encoded a tiny RNA, microRNA, that did not code for a protein. Ruvkun cloned the Lin14 gene, and the two scientists realized that the Lin4 microRNA sequence matched a complementary sequence in the Lin14 mRNA. After his postdoctoral research, Victor Ambros analyzed the Lin4 mutant in his newly established laboratory at Harvard University Methodical mapping allowed the cloning of the gene and led to an unexpected finding. The Lin4 gene produced an unusually short RNA molecule that lacked a code for protein production. These surprising results suggested that this small RNA from Lin4 was responsible for inhibiting Lin14. How might this work? Concurrently, Gary Ruvkun investigated the regulation of the Lin14 gene in his newly established laboratory at Massachusetts General Hospital and Harvard Medical School. Unlike how gene regulation was then known to function, Ruvkun showed that it is not the production of mRNA from Lin14 that is inhibited by Lin4. The regulation appeared to occur at a later stage in the process of gene expression through the shutdown of protein production. Experiments also revealed a segment in Lin14 mRNA that was necessary for its inhibition by Lin-4. The two laureates compared their findings, which resulted in a breakthrough discovery. The short Lin4 sequence matched complementary sequences in the critical segment of the Lin14 mRNA. Ambros and Ruvkun performed further experiments showing that the Lin4 microRNA turns off LAN14 by binding to the complementary sequences in its mRNA, blocking the production of Lin14 protein, a new principle of gene regulation mediated by a previously unknown type of RNA, microRNA, had been discovered. The results were published in 1993 into articles in the journal. So, the published results were initially met with almost deafening silence from the scientific community. Although the results were interesting, the unusual mechanism of gene regulation was considered a peculiarity of C. elegans, likely irrelevant to humans and other more complex animals. That perception changed in 2000 when Ruvkun's research group published their discovery of another microRNA encoded by the LET7 gene. Unlike Lin4, the LET7 gene was highly conserved and present throughout the animal kingdom. Ruvkun cloned LET7, a second gene encoding a microRNA. The gene is conserved in evolution, and it is now known that microRNA regulation is universal among multicellular organisms. In addition to the mapping of new microRNAs, experiments by several research groups elucidated the mechanisms of how microRNAs are produced and delivered to complementary target sequences in regulated mRNAs. The binding of microRNA leads to inhibition of protein synthesis or to mRNA degradation. Intriguingly, a single microRNA can regulate the expression of many different genes. And conversely, a single gene can be regulated by multiple microRNAs, thereby coordinating and fine-tuning entire networks of genes. Cellular machinery for producing functional microRNAs is also employed to produce other small RNA molecules in both plants and animals. For example, as a means of protecting plants against virus infections. Andrew Z. Fire and Craig C. Mello, awarded the Nobel Prize in 2006, described RNA interference, where specific mRNA molecules are inactivated by adding double-stranded RNA to cells. Ambros and Ruvkun's seminal discovery in the small worm C. elegans was unexpected and revealed a new dimension to gene regulation, essential for all complex life forms. The seminal discovery of microRNAs was unexpected and revealed a new dimension of gene regulation. The seminal discovery of microRNAs was unexpected and revealed a new dimension of gene regulation. Say good morning to our new medicine laureate. Gary Ruvkun was woken up in the early hours to the news he had been awarded the 2024 Nobel Prize in Physiology or Medicine. Stay tuned for our interview with him. Coming soon, congratulations to our 2024 Medicine Laureate Victor Ambros. He celebrated the news of his prize with his colleague and wife Rosalind Lee, who was also the first author on the 1993 Cell paper cited by the Nobel Committee. 2024 Nobel Prize Laureate in Physiology or Medicine. Victor Ambros was born in 1953 in Hanover, New Hampshire, USA. He is now Silverman Professor of Natural Science at the Amazon, Worcester, MA. This year's medicine laureate Gary Ruvkun was born in Berkeley, California, USA in 1952. He is now Professor of Genetics at Harvard Medical School.